this is how the adrenal gland would look under a scope. So you can see that we have the middle portion, which is, of course, our medulla. That's the part for the sympathetic nervous system. And then the zones, all right? So they based the zones on a lot of how it looks under the scope. So as you can see, we've got our zona glomerulosa, fasciculata, reticularis. And then, of course, this being a representation. And around, because when we look at organs, organs always have this like wrapping that's around them. I mean, they got to stay together, okay? So we've got a capsule, what they refer to as a capsule, that's going all the way around that adrenal gland. Now, even though we're seeing the medulla portion being with the sympathetic division and the cortex being mostly with our endocrine system, they're not independent. They actually depend on each other, the two parts. And their makeup is very different in the cellular makeup and so forth, but one stimulates the other. Without cortisol, the medulla would actually atrophy, which is not good. And then some of the cells of the medulla actually extend into the cortex. So they're very dependent on each other. When stress, once again, stress is not good, but it happens. When stress activates that sympathetic nervous system, the medulla cells activate the cortex to release their hormones. For example, the cortisol that we tend to associate with the stress response. And the one that they say, like with the stress and everything, makes us keep the belly fat. Y'all ever see the commercials on TV, like for the Relicore or whatever it's called, you know, and about how, oh, you know, cortisol and the stress reaction. Okay, so that's what they tend to associate with the stress response. Now, the pancreas, once again, we're going to have an organ that is associated with some different systems. Because we have the pancreas, it's exocrine and endocrine. For the exocrine portion, it secretes digestive juices, which are needed, of course, in the digestive system. For the endocrine portion, the endocrine cells are referred to as pancreatic islets. Simply means island. They're known as the islets of Langerhans, named after, of course, the person who discovered them. So, when you look at the pancreas under the scope, it looks like there's two different areas of cells. And so embedded throughout it looks like these little islands. Therefore, the term, the islets. These islets, if you look at their structure, they are said to have beta cells, alpha cells, delta cells. Their goal, secrete hormones to regulate glycemia. And glycemia refers to the blood glucose concentration. The glycemic index, I'm sure you've heard of that, okay? Now, those alpha and beta cells, and of course delta cells, they are very important in this function, and these are the cells that will get broken down or destroyed in the cases of diabetes. So diabetes 1, diabetes 2, we can look to this area of pancreas because the cells are usually damaged. Under normal conditions, the cells are going to respond to nutrient levels in the blood. Under normal conditions, the insulin gets released. The insulin is the key, so to speak, that unlocks the cell of our bodies to allow the glucose to be used. So that is how it should be normally. But we know that things can go wrong. And the result, diabetes. We tend to refer to diabetes as diabetes 1, diabetes 2. But we can't remember that we also have a diabetes insipidus, but that one is associated with ADH, which we'll talk about. So those alpha cells in the pancreas, in these little islands, their job between meals, if our blood glucose drops. Now let me explain real quick. The only, which I've mentioned before, the only nutrient the brain can use is glucose. So the body will do everything it can to provide the brain with glucose. Hopefully, if we eat a normal schedule, the body doesn't have to work too hard in between to supply the glucose. If it does, then it becomes issues of affecting the brain cells, which is not good. Usually some type of effects come from that. But under normal conditions, between meals, if the blood glucose level drops, the alpha cells of these little islets, these little islands, secrete glucagon. That promotes glycogenolysis and gluconeogenesis in the liver. This allows for the release of glucose into the blood so that the levels can rise. Glycogenolysis. What does that term tell us it's doing to glycogen?
It's breaking it apart.
Breaking it apart. Because the body stores extra sugars, the glucoses, and in short term, stores it as glycogen in the liver and the muscles. So in between meals, if we don't have enough glucose in the blood, it can simply take some of that glycogen in the liver that got stored, lyse means to break it, and give the glucose to the blood. But what about this gluconeogenesis that we mentioned earlier? Do what, Audrey? Makes a new glucose from stuff we really don't want it making a glucose from. Now, we wanted to use the fatty acids, great, okay? But that usually involves also sometimes breaking down of a protein, which we don't want. Okay, but it will do it if it has to. If we don't feed our bodies the right fuel and feed the fuel in a acceptable fashion, the body will do that. Not good all the time. Our beta cells in those little islands, they secrete my insulin and amylin. My insulin is the key that opens the cell to get it to use glucose correctly. Beta cells are usually, depending on which diabetes it is, are more affected based on the diabetes. For example, diabetes 1, okay? Diabetes 2 can be other issues coming along with it. But insulin is the hormone that we call the hormone of nutrient abundance. The goal, the norm, secrete it after a meal when hopefully we have consumed the proper nutrients. The target is my liver, skeletal muscle, and adipose tissue to absorb the glucose. Why? Well, the glucose that doesn't immediately get used, I can store it in the liver and the skeletal muscle in the form of glycogen, short term.
Plus,
Adipose tissue, I can take the short term and turn it into long term. So this is things that it's going to help with. It promotes synthesis of the glycogen that can get stored in the liver or the skeletal muscle, synthesis of the fat and protein that we can store and use. When it doesn't work or doesn't work correctly, it's known as the diabetes mellitus. I just mentioned a different type of diabetes. What was it? Diabetes what? Insipidus. So I have diabetes mellitus, which is for insulin. Diabetes insipidus is ADH, the antidiuretic hormone. Big difference. The term diabetes refers to urine. People tend to associate it with sugar, but it's urine. Mellitus lets me know it's the sugar portion. Insipidus lets me know it's the ADH portion. Big difference. Big difference. Amylin. When we eat, we need to make sure that the foodstuffs are not utilized or moved through our system too quickly. We need to make sure they get broken down properly, move through the intestines, continue to get broken down, and counter the very numerous blood vessels of the small intestine so that we can get those nutrients into the bloodstream and to the cells. So amylin's job helps slow down the emptying of the stomach so that the foodstuffs have time to get broken down and we can get the nutrients from them. The delta cells of that isolate, the island, they secrete somatostatin, which was my growth hormone inhibiting hormone. Does everybody remember that? So this one works with amylin, help us limit secretions of that stomach acid so that we can slow down the gut activity, meaning the stomach and the small intestines, so that we get the nutrients to the blood. That's the goal. But things go wrong, especially with those beta cells, the alpha and the beta. The gonads, the testes and the ovaries, they are considered to be both endocrine and exocrine. Once again, so exocrine is their product of either the egg or the sperm. The endocrine is going to